Welcome to INBIOSIS Webinar Lecture Series 1/2023. I am Sarahani, your moderator for today's webinar. The video that we played just now captures the gist background of our Institute of Systems Biology, located in Universiti Kebangsaan Malaysia, Bangi, Selangor. I hope it has given you a sufficient insight on the institute as a whole. So, for those who are interested to collaborate with us, you are most welcome to do so. Feel free to browse INBIOSIS website and also social media for the latest updates and also for some information regarding INBIOSIS. I feel so grateful today that all of us are granted good health and time to attend INBIOSIS webinar. This is our first webinar for this year. There will be more webinars to come, and I hope all of you will join us again in the future. Our honorable speaker for today is Prof. Dr. Zeti Azura Mohamed Hussein from INBIOSIS. Before we start, allow me share with you Prof. Dr. Zeti's research background. She is a professor in bioinformatics and computational system biology from Department of Applied Physics, Faculty of Science and Technology, UKM. She is also a principal research fellow at the Institute of System Biology, UKM. She obtained her PhD in bioinformatics from the University of Edinburgh, Scotland, United Kingdom, where her journey in bioinformatics started with structural bioinformatics. Since the last 13 years, her research focuses on how protein interaction network controls and regulate biological systems using computational approaches in understanding disease associations in human and also investigating plant systems to understand phenotype associations between proteins and the biological phenomena of interest. She and her team has developed a systematic method combining protein interactions, functions, and networks to an integrative analysis that is able to discover interesting modules which cannot be fully achieved using only a single type of data, which she will share with us today. Without further ado, let us welcome Prof. Zeti to share her experience with us. The screen is yours, Prof.
Thank you very much, Dr. Sarahani. Assalamualaikum and a very good afternoon to all. I would like to thank INBIOSIS webinar team for the invitation and also for arranging the session for me. Okay, I would like also to thank all attendees for spending your Friday afternoon to listen to my talk. And I hope that everyone will benefit from it and possibly will venture your research into network biology. So allow me to share my presentation. Okay. Okay. All right. So today I would like to share with you some progress work that have been carried out in my lab. And today I would like to talk about how we have developed a workflow to be used for protein interaction and also pathway analysis in order to identify new proteins or novel motifs and so on, and also to facilitate functional annotation. So as you can see, the title are as follow, and I will start my presentation with showing you these links and networks. So this is a network which has been central to our understanding of biological system. Okay, so they are visible in the earliest delineation of cells and cell connectivity, and all biological system, either in classical genetics, in the form of genetic linkages or linkages mapped among the genes as well as between genes and phenotypes. Okay, so this is the basis of our understanding of transcriptional circuits or molecular signaling, as well as metabolic pathways, and also the structure of the food webs and ecosystem, right? So, and then as you all are currently aware that the rise of social media has added to the new dimension to the study of biological networks. Particularly, social networks can be explicitly charted and quantified online. So basically, what we are doing in biology is similar to what is happening in our social network. Well, in the field of computational biology, for the last 20 years, have seen interest on networks have been growing from a curious sideshow to a conceptual framework for computation, where there are two views that have been developed in parallel to one another, which is a type of data as well as a conceptual framework. Okay, so meaning that the procedures and the method in analyzing this data. Okay, so as being the network as a concept, it proves to be very powerful, especially if you are looking at the huge number, huge amount of data that are available now from omics technology, and this data are supposed to be integrated. Okay, so the molecules in our systems, in the biological system, are interacting with each other, hence the importance of network analysis. Okay, so this is a yeast protein interaction network, which is the first published network study that has been carried out by Shwikowski et al., where they used this figure to illustrate a network of protein interaction in Saccharomyces cerevisiae, which has been generated from a global analysis of more than 2,000 published interaction between the proteins in the yeast. Okay, so as you can see here, this is just part of the network, and it contains about more than 2,000 interaction among more than 1,000 proteins. And then it is to show you that in the protein interaction networks, it is highly connected nodes. So these are the nodes, all right? And the linkage, the edges here illustrates the relationship between the nodes, okay? So from this work, they have shown that they have predicted correctly about 72% of the 1,000, more than 1,000 protein has at least one partner to another. So as you can see here, you can see, for example, this one have many interactions between them, but some only have one, and so on. Okay, so in general, by doing protein network analysis, we would be able to identify the linkages between all the macromolecules in the system. Okay, so let me introduce you to some of the the introduction of the network. So it is a set of ages, and then it is linked together. So for the last 5 years, we can see that many publication has been increased on the protein-protein interaction network, regulatory networks, and co-expression network, because people nowadays is post-genomics, and there is no longer work on the individual genes or proteins. So there are many more information that can be obtained from understanding this interaction. Okay. So this is a very powerful direction to be used, the knowledge of molecular networks to interpret the associations between genetic variants in diseases or probably in other biological system of interest. Okay, so it goes back, the complex network theory goes back way in 1736. It started with the graph theory developed by Leonhard Euler, which at the time focusing on using that method in mathematics. Okay, but the application of network in biology started in 1999 by Barabasi and Albert, which it also adopted from the network analysis that have been well established in studying economics, linguistics, medicine, social science, and etc. Okay. Right. So this is an example, the figure showing how we can conduct or start with a biological, establishing a biological network from a computational perspective. Okay, so the biological network can provide a conceptual and intuitive framework to investigate, to model, to characterize, and to understand complex interaction between the different components in the biological system. So now in the systems biology era, post-genomics era, we can see many omics data sets that have been produced individually, but somehow when you want to understand the biological background of those data, you need to link or you need to conduct an integrative analysis. Okay. Right. So in 2008, it has been proposed that this is the key to systems biology. So as you can see here, I've put up three examples of biological network where A is for transcriptional regulatory network, B is for protein-protein network, and C is for metabolic network. Okay, so the characterization of this network are similar with the one that we look in the internet network or World Wide Web, as well as society. Okay, so the transcriptional regulatory network here usually is conducted to display or to illustrate or to find out interesting properties that can be interpreted in a biological context for better understanding in the complex behavior of gene regulatory networks. Okay, so that control the whole system. And then the PPI or protein-protein interaction usually is used to describe various biological processes such as cell-cell communication, perception of environmental changes, protein transportation, and so on, which more onto the physical interaction. Okay, and meanwhile, the metabolic network can be used to understand the chemical reaction that catalyzed by different enzymes in producing essential components, as well as the use of energy that are necessary to synthesize and use them in constructing cellular components. So these are the so-called main types of biological network. So from the papers, published papers nowadays, we can see that there are individual there are findings on individual type of biological networks. But ideally, hopefully one day there is a work on trying to integrate all these pathways together, because those will give a good representation for the whole biological interest. Right, so here I introducing some of the words or terms that are widely used in network biology. So another one is interactome. So these are the example of the databases and approaches that that can be used to work or for you to start working in this area, as well as to get some information on what kind of database that you can use and what kind of method that you can use and so on. Right. So this is a slide here is to share with you what is the biological system. So in a simple word, biological system is a referring to the group of entities or organs or components that work together to perform a particular task or to conduct a particular task. So it could be in the neurological, it could be ecological, it could be molecular interaction, and so on. Okay, so this is where we want to know what are the components that are communicating with each other to ensure the proper function is executed. Okay, so from the point of view of a network, this is a component. A network consists of nodes and edges. So as you can see here, different networks implying or consisting of different nodes and edges. Okay, for example, in protein-protein interaction network, nodes are usually proteins and edges referring to physical interaction. Okay, So in a gene regulatory network, so there are gene network versus protein network. So those two are completely different. So for a gene network, usually it is used, or it is usually known as gene regulatory network, and it represents genes and lines between the regulatory interaction. And usually genetic networks are subject to evolutionary selection. Okay. Meanwhile, for the protein-protein interaction, the nodes are always represented by proteins, and the connecting lines are referring to the physical protein-protein interaction. For example, physical contact with high specificity established between the two nodes or the two proteins as a result of biochemical reaction that steer the interaction, which includes electrostatic forces, hydrogen bonding, hydrophobic effects, aliphatic interaction, and so on. Okay, so the genetic interaction capture functional relationship between the gene and phenotype readouts. Meanwhile, the protein-protein interaction identify physical connection between the proteins. Okay, so those two are complementary and they are usually non-overlapping. Okay, so by having those two, by conducting these two type of analysis, it enables us to see the global view of the functional architecture of the cell, which will, and also it will reveal the organization of the principle, and sometimes they are actually over evolutionary conserve. Okay, right. So, okay, in diseases. Okay, for example, using this pathway analysis, this study has able to identify the in-frame mutation that is enriched on the interaction interfaces on the proteins associated with the certain diseases. Okay, and then they also, from this study, they also have managed to predict about 229 candidate genes, and as well as they have managed to annotate a large number of unknown or previously unannotated proteins. So there is another motivation for us to use network biology to provide a new way of annotation, because as you can see, the numbers of hypothetical proteins are huge, and then sequence-based and structure-based analysis cannot are not able to satisfy or to complete some of the analysis. And another approach is by using pathway analysis or network biology. Okay, and then so next for the integrated network analysis. So first of all, how do you obtain biological network structures? So usually what we did is by retrieving these network interactions from databases, and then observing the network topology and look at the interaction directly from the high throughput experimental data. So you have two options on how you want to start off your biological network analysis. One, get your data from the databases, the published data, and secondly, you probably generate your own data, okay, from the omics. So these are the papers that you can read later to see how this work can be involve the mathematical modeling and so on. Okay, so how people use mathematical models on those data to predict the interactions. Okay, so now I would like to share what we have been doing for the last few years on developing a pipeline for network analysis. So this is a motivation based on the assumption that biological systems in nature are non-random, but they follow series of organizing manners, okay, in their structure and evolution. And and then most biological characteristics arise from complex interaction between the cell numerous constituent, right? So we have, when I first started doing pathway analysis, we were facing with difficulties in terms of referring the method. So there is no so-called standard method that can be used to construct a pathway or to start the pathway analysis. Okay, so then and then also we have trouble when we are using sequence similarity techniques and also sequence and structure-based analysis. We are not able to search for the missing genes or novel genes and so on. Okay, so that is one of the motivation. And then the second one is the complete annotation. So we dealing with a lot of hypothetical proteins, but using sequence similarity and structure-based sometimes is not complete. So we have to find another option. All right. And then, so these are the three main problems that we were solving during the way throughout this year to organize all these experimental data, and then to see how the pathway or the molecular components are interacting with each other, and to see how effective is the model that we built. Okay, so this is the overview, the common steps in molecular network. So it could be plant, it could be disease, and so on. Okay, so but in general, these are the so-called steps that are common for molecular network analysis. But somehow, please take note that this usually you need to customize your analysis, your pipeline based on the data type that you have. Okay, so but there is just a general common pathway that you common pipeline that you can use, but between those steps you need to add additional steps that are required based on the data that you have. Okay, so for the next few minutes, I will share with you several case study that have been from our lab. So I have put up here 4 case study. So in each of these case study, we are applying or we are using network biology approach. Okay, and from a different case study will give you provides a different type of result. So here I would like to give you some overview. For example, if you are using this kind of method, what are the results that you are expected? Okay, so there are 4 case studies. 4 case studies here. So then, for example, this one, this is where we the motivation is to search for the missing genes. Okay, so the data that we are using from this 4 case study are basically the first option, which comes from the published data from the published data from the databases as well as from the literature. So in this one, we use or our subject is on the glucosinolate. So we are looking for the glucosinolate genes. So as you can see here, I put up the method briefly because today I don't want to talk about the details of the results, but I would like to talk about or introduce the steps that that we have done to answer certain questions in the research. Okay, and also to give you with this kind of work what kind of result that we are expecting. Okay, so in here for this one we are using graph clustering approach on the data set GSL. GSL is abbreviation for glucosinolate, so it's GSL genes. So using graph clustering approach, we are calculating the clusters to see how many clusters on these genes, and then we conduct a statistical analysis as well as pathway enrichment analysis. So in the end, the results we have managed to identify several genes that have not been known previously. Right, so this is the flow chart for this first case study. So this is a step-by-step procedure to identify potential GSL genes involved in GSL biosynthetic pathway. So as you can see here, we are using databases, literatures, and so on to construct our data set. Okay, so once we have construct the data set, we conduct a gene co-expression network to see the interaction between all those data, and then do the clustering by using the graph clustering techniques, and then conduct the statistical analysis to see if the clusters are significant, and also to see if the p-value should be corrected because it is to increase the level of accuracy, okay, and also the reliability of the techniques, right? So this is the pathway for that until we manage to identify the potential GSL genes. Okay, so these are some of the results. So this is the example of the result where we managed to produce a gene, or we have produced a gene co-expression network. And then from here, as you can see here, the expression, the network was constructed from different databases. And then you can see that the network is developed using is developed from 270 nodes and 5,000 edges. And also from here, we managed to identify the function and also the molecular function of the proteins. Okay, all right. So throughout the process, here I would like to share with you that from the analysis, from the experiment, we managed to find several, about 13 proteins that have not been identified before. Okay, so for example, here BET5 and also GTR. Okay, so one of our hope is that I believe that every one of you are using CAC pathway, but note that CAC pathway is not complete, and it is developed, or it is constructed based on many data from many organisms. Okay, so and then that's why sometimes when you search into the CAC database, you are not able to identify your proteins or genes of interest. So by using this approach, we are able to do that, and we hope that by publishing this paper, it will contribute to the new to the update of the databases. Okay. Right, so these are some of the data that we have on how we manage to identify all the genes, the 13 genes from the clusters. Okay, so detail of the paper, detail of the work can be read into the paper. So then here, to show you that we can use network biology and pathway analysis to identify all the unknown proteins. Okay, so apart from that, we also conducted this pathway enrichment to see the functional module of these proteins and how they are interacting with each other and in with which category. Okay, so the pink nodes denote all the known GSL that encode for the enzymes, fiction, TFs, and also transporters. Meanwhile, the purple nodes for the newly identified potential GSL genes. Okay, so these genes, so what we did here is basically based on network analysis, but in order to see if they are truly function in nature, validation experiments must be carried out. Okay, so in the end, these are the findings: 13 potential GSL, 5 enriched clusters, and so on. Okay, so next we go to the next case study. So in this case study, this work is to illustrate that how network analysis can be used to illustrate or to determine the evolutionary relationship between the homologs. Okay, and if they can, if the function is sustained. Okay, so in this case, we are working on Arabidopsis and rice genome sequences. focusing on the sulfur-containing compound. And then why we are choosing sulfur-containing compound? Because it is important in plant defense against pathogen and diseases. Okay, in here we did a synteny analysis to see the relationship, the evolutionary relationship between the homologue on the chromosome. And then we conduct, as you can see, we conduct a multiple sequence alignment and phylogenetic analysis, which is a standard bioinformatics analysis. And then we include gene ontology, enrichment, and pathway, because we want to see how it is function in general on this case. Okay, so the results has given us the identification of some putative biosynthetic genes using synteny analysis, the results of the phylogenetic analysis, gene ontology, and so on, as well as chromosomal distribution of the synthetic SCC synthetic biosynthetic genes, where this information will be useful for the rice breeding project and mitigation. So this is an example of the results that we will obtain, showing that the interaction between genes, SCC genes in different organisms, between OS or Rosai sativa and Arabidopsis thaliana, and it has shown that there are about 89 SEC from Arabidopsis and 269 ECC from Oryza sativa. So they had, from here, there are, we found out there are 477 synthetic gene pairs that were represented by different colors. Okay. Right, so this is the result from the pathway analysis, where it shows you that the distribution of biological processes and molecular function, and then how it is shown between the Arabidopsis and rice autologous species. Okay, and then so as you can see here, the classification of the function of the proteins of the genes. Right, so as a conclusion for the finding from this kind of analysis, we found that our rice biosynthetic genes show synthetic association with Arabidopsis homologues, and then it has a high degree of conservation between the two, which could be implicated the function of SEC during defense response. And then for the, as I mentioned earlier, the chromosomal level mapping of SEC gene will be useful for functional research and breeding strategies. Okay. Right, for the case study, so as you can see, a different approach will include, which includes some pathway analysis, will give you different, is able to achieve the results that you are hoping for. Okay, so for the case study 3, is the association of polycystic ovarian syndrome data with the type 2 diabetes mellitus during, and so in general, what we wanted to do here, what we did here was to look at the association between PCOS and diabetes, and to see the effects of the drugs that have been used to control diabetes, whether it can be repurposed and used to manage PCOS. So in these two case study, these 4 case study, 2, as you can see, 2 comes from the plant-based work, and another 2 is from the human disease perspective. Okay, so from here, by using this, as you can see, the method here, as well as the results. So here I would like to go straight to the method that we are using. So this is the flow chart that we have developed to understand or to develop the association between the diseases and the drug, as well as on the target. Okay, so we managed to produce the drug target network that illustrates the association between diabetic drugs and PCOS. Okay, and the target, sorry, and the targets. So as you can see here, these are type of drugs on the color, and then you can see which drug affect which genes or proteins and so on. Okay. Right, so this one the pathway. So we managed to identify several pathways based on the proteins that been shared between or that been shared proteins and between PCOS and diabetes. Okay, so we are during the process we are searching for the shared proteins between PCOS and diabetes to see what are the proteins shared among them that contribute to the association. Okay, so these are because with this idea, we can postulate that the drugs used in diabetes can also work on the protein that being shared together. Okay. Right, so then this is the pathway between network of PCOS and diabetes. So it is my apologies because it is very small, but however the mapping here, as you can see here, the diabetes drug, diabetic drug that used in PCOS and so on. So the color represent the data set here and where they are located on the pathway. Okay, so this is an example of the holistic view on the network that you can observe and manipulate later on the if you want to do experimental or probably if you are able to do the modeling and simulation, so we can see which drugs we will affect which targets and so on. Okay. Right, so here I've highlighted several interaction derived from constructed PCOS diabetes network. So and this finding actually will be further analyzed. So as of now, this is what we have. So I am hoping to go further to probably on the structural aspect to see how the drugs and targets interacts and how it can use or target into different target drug targets. Okay, so from here, the findings are the association between PCOS and diabetes. So it will, the detail of it can be explained further from the drug target network as well as from the PCOS diabetes network. And then the association of one of the drugs in shared target suggests the possibility of it being repurposed as a medicine for PCOS, and the association of drug insulin to targets in PCOS and diabetes will provide different insight and the possible risk for developing PCOS in diabetes patient. Okay, and also, as well as a potential repurposing of insulin as an alternative treatment for PCOS. Okay, and then the last case study is the computational identification of potential PCOS biomarkers from protein disease interaction network. So again, these are the method and the results here. So here, as you can see, we have the specific data set here working on the pathway network and pathway construction and using two types of analysis, cluster analysis and hub gene analysis. And then in order to select the highly potential candidates, certain criteria must be developed to rank our potential biomarker candidates. So this is the PCOS putative pathway, and as is which has been generated from 199 sub-pathways that are directly involved in the pathobiology of PCOS. So basically what you can see here is the proteins that are related to PCOS. Okay, is a PCOS related proteins that are highly likely to play a significant role in the pathogenesis of PCOS. So from here, we have identified several or five major pathway that plays a significant role in PCOS, okay, so which is insulin, FOXO, and so on. Okay, so as you can see here, the red ones here are possible or potential PCOS biomarkers in different part of the pathways. Okay, so however, this is computationally predicted. Okay, so there are many steps that need to be carried out before we have highly reliable data sets for experimental validation. Okay, so as we did some criteria and development which applied on those pathways, so this in the end we managed to filter or choose the highest ranking candidates, which those are 28 candidates, and these are so-called potential biomarkers are experimentally worthy. So, okay, so meaning that I hope that I will get some funding to work on the experimental validation of these biomarkers. Okay, so for the findings, you can see here the shared pathways and also a list of potential biomarkers. So in summary, from different case studies that I presented just now, which use somehow in some steps or another, some pathway analysis and also protein-protein interaction or gene gene co-expression analysis. This is to demonstrate that the inclusion of network biology approach are able to identify, for example, in case 1, identify missing genes. In case number 2, we managed to identify or to understand the evolutionary relationship of homologs. In case number 3, understand the disease situation association and how the established drug can be repurposed. And in case 4, the identification of biomarker candidates. Okay, so these are an example of the application of network biology in your data set. Okay, but however, some people are asking, is there just one right method? So basically, in general, there is no right method in doing this kind of analysis, where you need to, in order, there is no, it depends on the type of data that you have, okay, as well of the size of the network and so on. So that is one of the reasons why we've come up to develop this kind of integrative analysis. Okay, so there is no one right method that fits all different biological, technical, and experimental design. So from years of our work, so we have we have developed this kind of guideline, a pipeline on how you want to conduct pathway analysis. So first step here, this is the gist of this study of this talk. So first step here, you need to look for the gene search. Okay, the second step is doing the protein-protein interaction or gene interaction, wherever you data set that you have, and then do the enrichment analysis. Only then step 4 for the pathway construction. So basically, this is I hope to have it as a standard guide on pathway construction or pathway analysis. Okay, so then once you search for, in order to search for the potential protein, you need to conduct a functional enrichment analysis and then do a pathway analysis using these, and then and include all the statistical analysis as well. Okay, so then, right. And the last one is once you get the curated genes and potential genes, so we use cytoscape a lot in our pathway integration, in also in data integration as well as on visualization. So when I started use cytoscape in 2009, there is a very basic with no more feature. It's just only for visualization. But nowadays, I think is cytoscape 13 probably, I can't remember the version. So it has improved a lot, and you have a lot of features that comes along to help you develop the not only visualizing but developing or extending your pathway, your network. Okay, right. So those are three so-called phase of the integrated pathway analysis. So I hope so. This work is currently will be published soon. So hopefully it is acceptable for publication in a good journal. Okay, so another tips for doing pathway analysis is, first of all, is you must know your data. Okay, so you need to know all the data, because by knowing it, only then you are able to choose what kind of method that you're using, what kind of database, and so on. Okay, and then know exactly what kind of analysis that you want to conduct. Usually it is guided by the research question. Okay, and then you must ensure your data is of high quality, because this is a so-called tagline in bioinformatics: rubbish in will produce rubbish out, and you obviously you will give unreliable results. Okay, and then as you know, there are many tools are available, so it is advisable not to use only one tool. So you have to use several tools and compare the results, and then note all your analysis type and the details. And then another thing, important thing that I realized is that you need to conduct a statistical analysis because it is prediction. And then do your statistical analysis with adjusted p-value. Okay, not the nominal p-value, because nominal p-value usually lead to the false positive or inconvincing prediction. Okay, so you need to do the adjusted p-value. So, and also a different statistical technique will generate different results. So you need to use several to see which one suits provide the expected results. Okay, and then visualization techniques is also important, because of the data. You need to see the data holistically, so you need to be able. So for the pathway analysis, we highly recommended using Cytoscape, because it has many features that can help you to how to say, to play around with your data, with your pathways. Okay, and then finally, you need to verify your results, whether with recent literature and database, or with the expert, with the biologists. Okay, because you need to avoid the problem of outdated annotation. And then the because throughout my experience, I think that it's very helpful to have an opinion from the biologist or the data producer, but because it gives you a different view and perspective on the biological context of your data. Okay. And then another take-home note is that no matter how efficient and robust a computation method that you are using, to have meaningful result, you must have a good quality data. So we are usually spending some significant amount of time cleaning up our data. Okay, so there are many ways of how to clean up your data, depends on the type of data that you have. Okay. So the work or the I have been working with all these great students of mine, and now they are all independent and independent scientists. So they have been trained to work on network biology. Okay, so and each of them have their own strength on protein interaction, gene co-expression, and so on. And the work is materialized due to their commitment and dedication. And then feel free to communicate with them if you think that they can help you in your research. You can collaborate with them. And unfortunately, we don't have Dr. Shadila with us any longer because she is now working in Japan. So, but she just the figure that I presented just now on the method was illustrated by Dr. Shatila. Okay, and then this work have been funded by various grants locally and also internally. Okay, so that's all for today. I hope somehow the information shared today will be beneficial, and if you have any question, please feel free to contact me via email. Inshallah, I will be able to help and have a fruitful discussion with you. Thank you very much for your time, and I'm sorry if my presentation is a bit too much or it may be overwhelmed because of many different kind of data set that we presented just now. Okay, thank you.
Okay, thank you very much, Prof. Dr. Zeti, for sharing with us very interesting topic. Now, I would like to open the floor for the question and answer session. Everyone, please type your question in the chat box and I will read the questions for you. Actually, we have some questions already in the chat box. I will start reading the questions now. So first come from Vinotieni. So a question is, in your opinion, what is the future directions of network biology in systems biology studies?
Okay, all right. So that is the way to go. So you need to it has been increasingly used in systems biology. Actually, it complements the systems biology study. Okay, so if you are familiar with the data produced from the systems biology, in the end, in order to integrate them and to see the holistic, the whole system holistically, you must apply network biology. So I think it is very, if it is a suggestion to anyone who's working in systems biology, should somehow incorporate the network biology analysis because you need to see the interactions and communication between those molecular components from different hierarchical levels. Yes. Okay.
Okay. Another question by Dr. Go. What are the challenges of network analysis?
Okay, so the challenges of network analysis, first is dealing with the data. Data itself, where you need to prepare your data. One, because those data have different formats and different. So you need to process your data to be. One is data processing lah, preparing a good quality data. Secondly is the selection of method that you analysis method that you need to use need to identify for the network analysis, and then the databases as well. So if so while doing network analysis, we sometimes depends a lot on the interaction databases, okay, that has published interaction data. Okay, so we rely on that one as well. So for that, we require that those data will be updated, but somehow it is not. So you have to do the literature searching looking for, because sometimes they published in the literature but it's not updated in the database. So those are the challenges. So usually one, and then another one is you must know the research question because it will lead you to the experimental design. Okay, because I think that throughout the experience, the important thing is to have your research question clear. Okay, only then you can design the steps and the steps for the next analysis. Okay.
Yes, of course. There are also some limitation, right, Prof, when we are studying species that are not included in the
Mm.
databases like string, right? So we need to do
Yes.
computational analysis on that.
Yep. Mm.
Yeah. So another one by Zaino, she's asking whether you can share your slides afterwards
Okay,
because he's
can. Okay.
interested in annotating the
Okay.
hypothetical and putative genes. All
Okay.
right. So another one by Dr. Moni. So she wants to know, uh, she wants to know if the proteins or genes that showed in the results are hypothetical or functionally characterized.
Okay. For our purpose, we were using functionally characterized. The
Yes.
one that we presented on the, you can see the different types of pathways, right? So
Mm-hmm.
all pathways uh, were generated using characterized protein. Okay, because during our data and processing, data preparing, we omit the hypothetical proteins. Okay, and then there's another work on the annotation of hypothetical protein using network analysis. Okay, so the one that you that I've shown just now are the proteins that are functionally characterized, meaning that
Yes.
known proteins. Yes.
All right. So another one by Dr. Moni, she's asking the differences between hypothetical data and putative data.
Okay. So tu Dr. Sahari boleh jawab
The IPM.
So hypothetical protein is unknown protein lah. Putative data means that the function is so-called interpreted from the computational analysis. Okay. Yeah. For example, you ada putative protein A. Okay, ada function A. So it is putative but dia ada similarity with the protein A tapi computationally, bukan experimentally. Okay, so I hope it's clear.
Yeah. All right. So another one, how do we annotate the protein or compound in the case of omics data? Is there any guidelines that should be followed instead of the database library, particularly for unknown compounds?
Okay. So, for example, if you generate omics data, usually each omics platform has their own database library, right? So you have to, first step, you have to search against the library. So just in case you don't have, the result is not significant or no results, then you can use computational using other database or literature, depends on what kind of data that you have. So in terms of proteins, you can do a structure based, sequence based, or network based. And if it is a compound, currently there is a method that you can use structure based or structure similarity search to identify the compound obtained from metabolomic study. Yes, there is available. There is some published work on that one too.
All right. Okay. Another question from
Yes.
Faris. What are the tips that you can share if we are doing pathway analysis on non-model organism?
Pathway analysis and
Faris.
non-model.
Yeah.
Non-model, what kind of tips you want to know? Doing pathway analysis on non-model. So it depends. Either you want to do protein, it doesn't matter. It is not necessarily have, you need to have, how to say, uh, okay. You it is not necessary to have a reference organism to start working on pathway analysis. Okay. Usually for a pathway analysis, you can start the work if you have a list of proteins. Okay. So once you have your list of proteins, you can do annotation. You can look for the annotation. And if those proteins are well characterized, it is quite straightforward because you can search into the, for example, UniProt database that has a detailed description, and you can look through the description, and in the end of the description and notation, there is information on the pathway. So you can start building your data or the apa punya, the information to before you build the whole pathway. Okay, so it doesn't matter if your data comes from a non-model organism or organism, as long as you have a list of proteins from that particular organism. Then
Yes.
only you can build up your pathway based on the previous results, based on the result from your previous analysis. Okay.
Okay, I think another question by Chin Ching here is also asking about the challenges using non-model organism. So she's asking how can we make use of the public database of our model organism and incorporate them in our data using the network analysis?
It's presented using model.
Because the question is in relation
Okay.
related.
Okay, lebih kurang lah related to Paris.
Yeah.
Yeah, so you probably need to look at your protein list lah. Look at your protein list or gene list, see if there is similarity uh, to your protein, to your data. So for example, if most of your data turns out to be hypothetical or unknown, you need to conduct a basic bioinformatic analysis first. Okay.
Yeah.
To help you annotate. So, for example, you have about 70% of hypothetical proteins in your data set. So what you need to do is put aside your intention to do pathway analysis straight away, but you have to annotate your hypothetical protein first, or hypothetical data. Okay, so by annotation, then only you can proceed to pathway analysis. So that is why one of the motivation for us to do that is, if you remember just now, is incomplete annotation. So you need to have a complete and reliable annotation before you can conduct pathway analysis. Okay.
Yeah, I think what we can help also if Prof Zeti mentioned about the basic bioinformatic, and we can do domain or motif analysis from there because we have the sequences, right? And then we can infer on the non-model organism based
Yeah.
on that information that we
Yeah.
have. So we
Yes.
can compare with the model organism.
Yes.
Yeah, right.
Another thing that I should mention is that this pathway analysis is categorized as an advanced bioinformatics. Okay, so and the BLAS MSA phylogenetic is a standard bioinformatics analysis. Okay, so somehow so that's why you need to know your data. Okay, so some data requires you to do the standard bioinformatics first before you proceed to this the advanced bioinformatics analysis. Okay, so
Yes, Prof.
you cannot leave out the basic bioinformatics analysis when you are doing pathway analysis.
Yeah, great. So we have another two questions here from Kashikan. Hi, Prof. How do we measure that our data is high quality enough for data analysis?
Okay, so how do you measure our data is high quality enough? Okay, so it depends on your data type, whether you need to so there are some parameters or apa punya characters of the data. It depends on the type of data. So for example, if you're talking about protein, you have to have your protein for example, full-length protein, and then it has the description, the biological description of the date of the protein and so on. Okay, but so those are the data that I'm referring from the published sequence from the database and so on. But if you are generating your own data in the lab, you have your set of parameters that you can use uh, to measure the quality of your data. Okay. For example, if you are producing or generating 3D model of the protein, you have certain analysis that you need to conduct to check for the quality validation. Okay, for example, look out for the Ramachandran plot, looking at the interaction the interaction between carbons and so on. Okay, so those are the type of quality checkup parameters. Okay, so I cannot be specific enough because I don't know what kind of data that you have.
All right. Okay, we have for one last question from Dr. Nisha. In the realm of multidisciplinary research, should one fear of computational biology? Please comment, Prof.
Big no. Yeah. So you need to apa panggil? You need to add this skill into your in your research. Okay, this is a value added skill. So I do encourage for the experimentalists or the biologists to have some kind of skill, computational skill to analyze your data. So it is not the time for you to rely on the bioinformatician is apa panggil is towards the end. Now some of the analysis, the computational analysis, you need to carry out yourself. Because why? Because in interpreting the results from computational analysis, it requires the knowledge, the biological knowledge, which somehow this biological knowledge is best described by the biologists themselves. Okay, so we bioinformatician, one of the challenges of being a bioinformatician is that when the time comes for us to interpret the computational analysis results, we are not able to fully describe or infer the analysis because we have a limited biological knowledge on that data. Okay, and that's why we require help from the biologists. So that is one of the tips that I listed just now, right? To go back to the literature as well as to go back to the biologists, the best to in order for you to accurately interpret the computational analysis results. Okay, and then answering to that question, I think one should not fear. So you must embark into some kind of computational analysis that are relevant to your work.
All right.
Okay.
Thank you, Prof. I think that would be the last question for today.
Okay.
I
Yeah.
must have a lot more questions to ask, so maybe we can have a coffee later on.
Sure, sure.
Okay, so actually we have come to the end of our webinar. I would like to thank Prof. Dr. Zeti for the very interesting research topic, and I'm sure that it has opened up our view and given us an idea of how we can bring up our research output to the next level. I would also like to thank all participants for spending your precious time with us today. There are a total of 122 participants for today, where 102 are from WebEx and 20 from Facebook.